Okay, so hello and welcome. My name's Daniel, and I've been working with the Struck with Daniel Lab on a project entitled Gene Level Analysis of Breast Cancer Heritability in European Populations. Next slide, please. So to start with, why study breast cancer in the first place? Well, breast cancer is the most common cancer in women in the UK and the leading cause of cancer death in women worldwide. Working out which genes are associated with increased risk of breast cancer could help us understand its causes and provide new opportunities for screening and treatment. Next slide, please. To explain the background of my project, I'll review 4 key concepts. Firstly, heritability. Heritability tells us how much of the variation that we see in a particular trait is due to genetic variation. The next concept is germline and somatic mutations. Here, we're investigating germline variants, which are those that are present at birth. Next is frequency and penetrance. You've probably heard of BRCA1 and 2. Germline mutations in certain genes like these significantly increase breast cancer risk, which is what we mean by high penetrance. However, these inherited mutations are rare or low frequency. In the broader population, breast cancer heritability could be explained by high frequency but low penetrance variants like SNPs. This takes us to our next concept, GWAS. GWAS studies have identified SNPs that increase risk, but most heritability still remains unexplained. A possible explanation for this is the polygenic architecture of breast cancer. Many SNPs with subtle effects contribute synergistically to increased risk. However, SNPs with weak effects are often discarded during statistical correction. Thus, we need a more powerful approach to investigate cancer genetics. Next slide, please. So how do we design a study to address this need? Let's talk through it with a schematic of the method, starting with our input data. We took GWAS summary statistics from 2 cohorts that investigated breast cancer, the Breast Cancer Association Consortium and the UK Biobank. For the SNPs in these datasets, we added LD scores and annotated them to genes. We then used a fascinating new tool called Baggara to estimate the contribution of all genes to breast cancer heritability. The output of this analysis was a list of significant genes from each cohort, which we then went on to characterize. Please press next to the animation. Now's a good time to mention the amazing training and skills I gained along the way, from programming and high-performance computing to data science and statistics. Next slide, please. So what did we find? On this slide, we have adapted Manhattan plots for our 2 cohorts. Each point on these diagrams represents a single gene. On the x-axis, these genes are organized by chromosome, and the y-axis tells us their contribution to heritability. We identified 631 significant genes in our BCAT cohort and 281 in our UK Biobank cohort, of which 108 overlapped. Next slide, please. Once we'd found our genes, it was time for some functional characterization to describe what we found. First, we looked to the literature for our top-scoring genes. Some genes were associated with breast cancer risk through GWAS studies or are involved in cancer-related processes. However, the real value of Baggara analysis is that we could use the entire gene list produced to perform more powerful characterization, such as gene set enrichment analyses and pathway analyses. These revealed significantly enriched terms that related to cancer in the literature. We also investigated overlap with cancer driver gene panels to identify links between germline and somatic mutations, but these were non-significant. Next slide, please. So to summarize, I ran a bioinformatics analysis using GWAS data, curated a map of breast cancer risk, and performed some functional characterization. Future research could build on these results by exploring breast cancer subtypes and mapping to other datasets. Next slide, please. Finally, I want to say an enormous thank you to my incredible supervisors and the Genetics Society for their support. Thank you.